Hello, Assalamualaikum. We are from Group 13. We'll present the assignment of Biochemistry and Biomolecular Technique with the given topic is Pentose phosphate pathway.
Hello everyone, I'm Revati Paramasivam. Today I'm gonna talk about the introduction of pentose phosphate pathway. The pentose phosphate pathway, defined as the metabolic pathway parallel to glycolysis, which generate NADPH and pentose, which is the 5-carbon sugars, as well as ribose 5-phosphate. The pathway is also known as the phosphogluconate pathway or hexone monophosphate shunt (HMB). The primary role is anabolic as it involves oxidation of glucose. It occurs in cytoplasm of both prokaryote and eukaryote. The pentose phosphate pathway began with glucose and has multiplied of reaction.
My name is Nadia Natasha binti Idrus and I will talk about mechanism in pentose phosphate pathway. There are two phases in pentose phosphate pathway, which is oxidative phase and non-oxidative phase. The oxidative phase is irreversible while the non-oxidative phase is reversible reaction. Pentose phosphate pathway is a reaction of glucose 6-phosphate plus 2NAD plus water will produce ribose 5-phosphate plus 2NADPH plus CO2 plus 2H plus, which the main product from pentose phosphate pathway is NADPH and ribose 5-phosphate. Oxidative phase will produce NADPH, which NADPH effect in reducing power in anabolic reaction and maintain the store of antioxidant inside the body. What is the difference between NADH and NADPH? NADH is produced in cellular respiration during the breakdown of glucose. The difference between these two is in relative amount of the reduced form and the oxidized form inside the cell. NAD plus will accept the electron in breaking down the glucose and produce NADH, while NADPH will donating the electron. NADPH is important in anabolic reaction, which anabolic reaction involves in building up the molecules, as for example is in synthesis of fatty acid. There are two steps in phase 1, which is the first step is dehydrogenation of glucose 6-phosphate to produce 6-phosphogluconate. Glucose 6-phosphate was oxidized to form lactone and NADPH is produced as a byproduct. NADP plus reduced as NADP plus that superscript plus N superscript and reduced as glucose 6-phosphate in the process of oxidation of glucose 6-phosphate. The water is used to form 6-phosphogluconate, which is 6-phosphogluconate is a lactone. The enzyme used in this process is gluconolactonase. The next step is hydrolysis of 6-phosphogluconate and decarboxylations. Hydrolysis happens for a lactone, which addition of water is to cleave a bond since lactone is intramolecular. The enzyme 6-phosphogluconate dehydrogenase is used to perform oxidative decarboxylations, which converting the 6-carbon skeleton to a pentose. Carbon is removed and release CO2. The electron released from the cleavage is used to reduce NADP plus to NADPH. The decarboxylation produces the formations of ketone, which is ribulose 5-phosphate.
Hi, I am Rishdan and I am talking about non-oxidative phase. Ribulose 5-phosphate is the substrate for two enzymes, which is ribose 5-phosphate ketoisomerase, which ribulose 5-phosphate to the corresponding ribose 5-phosphate used for nucleotide and nucleic acid synthesis. Ribulose 5-phosphate 3 epimerase, which alters the configuration about carbon, giving cellulose 5-phosphate. For the reaction, cellulose 5-phosphate with 5 carbon and ribose 5-phosphate with 5 carbon reacts to give glyceraldehyde 3-phosphate with 3 carbon and pseudoheptulose 7-phosphate with 7 carbon as result by the enzyme transketolase. Glyceraldehyde 3-phosphate with a 3 carbon and pseudoheptulose 7-phosphate with a 7 carbon is acted by transaldolase to give fructose 6-phosphate with a 6 carbon and erythrose 4-phosphate with 4 carbon. Erythrose 4-phosphate with 4 carbon and cellulose 5-phosphate 5 carbon reacts in the presence of enzyme transketolase to give fructose 6-phosphate with 6 carbon and glyceraldehyde 3-phosphate with 3 carbon. Magnesium 2 plus and thiamine diphosphate, vitamin B1 as coenzyme. Subsequently, fructose 6-phosphate is isomerized to glucose 6-phosphate by enzyme phosphohexose isomerase. Glyceraldehyde 3-phosphate exists to make a reversal of glycolysis, and the glucoogenic enzyme fructose 1,6-biphosphate or its process to glycolysis.
I am Muhammad Hafiz Leong and I will summarize for the topic pentose phosphate pathway. The pentose phosphate pathway oxidizes glucose to make an ADPH and other carbohydrate for biosynthesis. For the first phase of the pentose phosphate pathway, it generates two NADPH molecule and a pentose sugar called ribose 5-phosphate. The major route for reduction of NADP to NADPH is the reaction of glucose 6-phosphate through two successive reactions. In the first reaction, carbon 1 of glucose is oxidized by glucose 6-phosphate dehydrogenase. In the second reaction, the same carbon is further oxidized to carbon dioxide and released, leaving behind a 5-carbon sugar in a reaction catalyzed by enzyme 6-phosphoglucolectin dehydrogenase. This is the second phase of the pentose phosphate pathway. The overall net reaction of this phase transforms ribose 5-phosphate molecule into fructose 6-phosphate and glyceraldehyde 3-phosphate. These glycolytic intermediates can be transformed back into glucose 6-phosphate, which in turn can be oxidized to form additional NADPH molecule. The non-oxidative phase links the glycolytic pathway to the pentose phosphate pathway, and it allows our cells a way to break down ribose molecule ingested in our body.